think that it should be a class.
Yeah.
Okay.
I should explain more.
So this is something you should think about before you collect your, oh, actually, just generally, and I probably already spoiled this, if I tell you, to remember my train of thought here. Right, so generally when I say I want you to have the most power you can have, what do you think the solution to that problem is? Collect as much data as you can. But this is focusing on, or actually, this is where I was going. What you should do before is do a power analysis like what JB was just saying. But almost all the time, you are not going to have pilot data because you need that mean estimate, and even worse, the variance estimate. So if you don't even have pilot data, this talk is for you. Because you can still get an idea. You can't estimate power, but you can do things to help get the most power out of the subjects that you do have. And we already
Is there a rule of thumb to know how many subjects to have in the pilot group?
There's never a rule of thumb. Enough. More than 3? That's the, and a tricky thing, I'll just say this about pilot data, is technically, let's say you collect 20 subjects for pilot data, then you cannot combine those with your, let's say you collect 20 subjects, you run a power analysis, the power analysis says you need 50 subjects, you can't just collect 30 more subjects and lump it together. You actually have to collect 50 new subjects. That's because if you want to understand why, sample size is assumed to be fixed. But as soon as you allow your sample size to depend on data, you have now made your sample size random. So your sample size is itself, it's a random thing, has variance associated with it, blah, 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 and it ends up inflating your type 1 error rate if you do that. So it's a tricky situation for many reasons. Your pilot study, although you want your pilot study to, the design that you use to match your future study, it never does. I've seen enough pilots run. So many things go wrong when you run your pilot. And that's actually the point of the pilot for me is more just to make sure the scanning protocol is going to work, your subjects aren't freaking out in the scanner. You can look at motion, and if the motion goes crazy at the end of the run, then maybe your run is too long, things like that. So it really is hard to have good pilot data for power. So we're going to go back to not having pilot data and thinking about what we can do to increase our power. So we're stuck. The NIH has only given us enough time and money to collect 20 subjects in our clinical population, which is 20 is awful, but life. I know that this is what happens. So a lot of people, when I say things like, if you want to look at between subject differences, if you want to look at correlations between bold activation and behavioral measures, I would be happiest if you had at least 90 subjects. So I gave a talk like this, and I heard after I gave the talk that the clinical people they gasped when I said it, but I couldn't hear it, and I get it. So I want to give those people something that they can do. You can never make a sample size of 20 behave like a sample size of 80. And I don't know what the answer is until we have more money for our studies, blah, 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 blah. So
we do collective collaboration and we collect data by the site. So.
Yeah, or people get clever. I know someone who with an R21 has an N of 100 in each group on an R21. Because she's working with other people and is very lucky. But I don't think that type of effort would have been made if there wasn't such attention drawn to our sample size problems. Okay, so I'm going to zero in on that variance that JB was just showing a little bit closer. And this is the only equation in here. And then I'm going to show you in that variance, there are 3 things we don't know, or 2 things we don't know, but 1 thing we do know, even if we don't have data. And we're going to focus on that thing that we do know and that we can control before we collect our data in a way that we can lower the variance. If I lower the variance, I increase my power. Does that make sense from what? I'm so glad you just did that because it was in the denominator. Right. So how many of you have analyzed fMRI data before? And this is fMRI specific, right? So what you do your first level analysis, and then you do your second level analysis. And we do that because it's our easy way of doing a mixed model. So what variance do we get at the first level, the stage 1, the within subject model? We have a within subject variance estimate from that model. And when we do our second level model, that's our between subject model, and we have a second variance component, the between subject variance. So depending on what software you're using, these things are estimated separately or not. But I'm going to work with the full definition, not the simplified assumption. But my point is we have these two variance components. And the way those show up in the overall variance is this equation. So we run a mixed effects model. So MFX just stands for mixed effects. And, okay, this term right here, all together, this is that level 1 variance. So the overall variance is the sum of two things. The level 1 variance component, or the contribution, and then the level 2 variance contribution. Okay? So in this equation, that's our between subject variance, which I'm assuming we don't have pilot data, so it's probably not known. And if you don't have a lot of pilot data, you're not going to be able to estimate that very well anyway. And again, I can't tell you how many you need. This within subject variance, that's probably not known either. But this bit, which I guess I should have explained, if we know what design we're going to use, we can create this. So all this is, so most of you have run a first level before. This is the design matrix for your first level analysis, and this is the contrast for your effect of interest. So if your contrast is faces versus houses, that'll be a 1 minus 1 contrast, and your design matrix will have a faces regressor and a houses regressor. And this is just your design matrix transposed, then multiplied by itself, the inverse of that. I think I pull that apart. Oh. So here's an example of a design matrix. This is FSL, and a contrast. So this is a stop signal task. So these are the go trials. This is the regressor for go. This is the regressor for the successful stop trials. This is the fail stop trials. And this is junk. The subjects just didn't do anything, but we're going to model it. And the contrast of interest is successful stop greater than fail stop. So this is the contrast. So X is this upper portion, and C is this vector down here. You've all seen that before? So our goal, again, is to make this variance as small as we can. So since we don't know these two chunks, we're going to do that by making this as small as we can. Or if I flip that thing over, I would want the inverse of it to be big. And that's what efficiency is. It's just taking that one part of the mixed effects variance and inverting it. And so now our goal is to minimize variance or maximize efficiency. Yep. So this is the most frustrating thing, and I gave a similar lecture at a different summer course a couple of weeks ago, and even though during the lecture I said this, during the lab, people were asking me if their efficiency was good. They said, I got an efficiency of 3, is that good? And that's the most frustrating thing about efficiency. I can't give you a cutoff for efficiency like I can with power. With power, if you're above 80, 90%, that's good. But all efficiency allows you to do is rank your designs against each other. But that's still a very useful tool, and you can look at how much the efficiency differs from two models. You can use that to see actually how much the variance is changing, at least that within subject chunk. So you use this as a ranking tool. The thing you have to be careful about is you might be in a situation where you're ranking junk and you're picking the best piece of junk out of all your junk. So I'll give you a tool to make sure you're not sifting through a bunch of junky designs. And that's where this variance inflation factor metric comes into play. All right, so it's just a relative measure. So this is an illustration where I'm going to take And what you do to generate different designs, I'll just say this really quickly. Different things will impact your efficiency. The orderings of your trials, so let's say you have positive, negative, neutral stimuli, the orderings of those will change your efficiency. What's the most efficient type of design you can have in an fMRI experiment? Yeah, the boring block design, right? So, and you'll see when I look at it later, I let it randomly order the trials and find the most efficient one, and the blockiest one ends up being chosen. So all you have to do is you have to put restrictions on it so it doesn't make it too blocky. So one thing you can do is you can block things a little bit, but not too much, and that'll make it efficient. Another thing that affects efficiency, or another thing that hurts, that makes a design less efficient, is when your stimuli are too close to each other. So if you increase the amount of fixation between your stimuli, that can really help. Or if you jitter the amount of fixation between your trials, that can help as well. So trial order, jittering, changing the ISI, all these things can help. And you basically change those and you estimate these ranks, and then you rank your designs and you pick the best ones. So that's the sneak preview into what we're going to do, and that's what the code I'm going to show you does. So this illustration, what I did was I wondered to myself, what if I took the least efficient design and the most efficient design, what would the power look like? But of course, I can't compute power without having those two variance terms. And what's the third piece of information I need for power? The sample size? Okay, there's more. What else would I need? What? Okay, what else would I need? I lied about there being one more thing, obviously. The size of the effect, right? The mu from JB's talk. So the sigma is now actually 2 sigmas in the X transpose X thing, but I also need the mu. So what I did was I estimated sigmas using real data, and hopefully this is a realistic illustration. I think I used that stop signal task, but then I don't know if the design I used is realistic, but I think this would hold. The design I used was assuming 2 stimuli, just positive and negative images. The stimulus duration is 1 second long. The interstimulus interval is fixed at 2 seconds, which generally I don't use fixed, but this is just a illustration. And the contrast of interest is positive versus negative. So hopefully you're picturing the design matrix that's going to have one regressor for positive, one for negative, and then the contrast will be 1 minus 1. All right, so that's what the X transpose X part looks like. And I'm going to do a search by just randomly ordering when the positive and negative stimuli occur. Let me, whoopsie, as a reminder. Is this important? Oh. Okay. So I'm going to do my search to find the best and worst design based on this value. These 2 values I estimated from real data. So this isn't something you could do in real life. But the reason I'm doing this is to show how much the power is going to vary from the best to the worst design. And the delta, or the mu, the effect size, I'm going to vary this. So we're going to get a power as a function of effect size, and I'm going to fix the sample size. I just wrote that where like one person can see it. Sorry. This just says mu varies. You'll see it. I have it. Okay, so this is just repeating what I just said. So what I got from the real data, and these aren't percent signal change units because I didn't really care. The within subject variance was 3,036. The between subject variance is 810. So, I don't remember how I selected the voxels. I will say in this example, for these data, the between subject variance is quite a bit smaller than the within subject variance. I'm not sure that's always going to hold for different studies, so this might not hold in general. But when this is the case, when this between subject variance, when this part is so small and this part is so big, this becomes more important, because this is effectively going to shrink this component. But if this component's already really small compared to this, it won't matter. But in this case, this is really small, this is really big, so changing this is going to change this quite a bit. Said another way, if the between subject variance dominates, this will matter less than this example. N is 30, alpha is 0.05. And again, I'm going to look at the power as a function of effect size. So the mu from JB's example will be my x-axis, power will be the y-axis. So the most efficient design, the power curve's in orange, and the least is in blue. And it does make a difference. So here, for an effect size of around 24 or so, there's almost a 20% difference in power. So whether this is a realistic, I think it's realistic example, it's just to show that even when you can't do power, this could be having an impact on your end power, even though you can't compute it. It's worth doing. So, I said this earlier, things you can do to try to increase efficiency is do random trial orders, jittering your ITI, increasing your ITI, lengthening your stimuli often helps, and increasing the number of stimuli also helps. And now I'm going to, I should have mentioned this earlier. When you do this, it should be when you're doing your behavioral pilot. You shouldn't do your behavioral pilot first and then say, okay, I'm ready to I got a beautiful behavioral effect. I'm now going to put this in the scanner. Jeanette, will you do an efficiency calculation for me? And this happened to me recently, and unfortunately it was with a graduate student's project who didn't have a lot of time to finish. And the problem with the design that was used in the behavioral pilot is there was each trial was, say, 10 seconds long, and the trial had about 4 different things that happened. But there was no fixation between. It was like something shown, the subject does something, something else happens, and then the subject's told to do something else. And they were unfortunately wanting to look at this effect, this effect, this effect, and this effect all separately. And what I had to tell them was that you could either just only look at this whole block altogether, or we're going to have to start putting fixation between these to break this up. And can you tell me why for fMRI we can't have things so close? Yeah, the hemodynamic response ruins everything. Our signal gets filtered through that, and it makes it sluggish, and it makes everything run into each other. And that causes correlation between our regressors, and correlated regressors are bad. That is what hurts efficiency. So for this design, we were very limited in what we could do without the worry of losing the behavioral effect that was there. So do it before you do your behavioral pilot. Yes?
Sorry if this is a silly question.
Don't ever apologize. Just ask your silly question.
Would any of these affect the within or between subject variability?
Oh, would changing these affect changing these will change this part right here. Whether or not it'll affect this, theoretically, no. But I suppose if spacing your stimuli out somehow made the effects more variable between subjects, I suppose that could happen. You know what I'm saying? Actually, that's exactly what happened in this example I told you. There was no jittering, there was no ISI, and I did have to change, fixation. And when I changed it, the behavioral effect did go away. So I'm sure the between subject variability did increase. But
also, your within subject variability should decrease, but not, it's not going to help you.
Well, not, this is based on the residual.
Because they're correlated now, so that they're like added smaller.
Huh?
The responses are correlated, so
But that's only impacting this part.
Okay.
Anyway, it shouldn't. Other questions? Okay. So this is just an illustration. Here is the unconvolved design matrix. So times zeros at top, and it increases as you go down. We have our negative stimuli in red, blue is positive. So of course, if we're modeling it this way, who cares about efficiency? We're fine. But in reality, we have to convolve that with the double gamma HRF. Don't cheat and use the single gamma. I know it inflates your effect sizes. Did you know that happens if you use a single gamma, which unfortunately is the default in FSL? Yeah, always use a double gamma, because the single gamma doesn't have a post-stimulus undershoot, so anyway, double gamma. So here's our design matrix. This is X.
You could write a PNS paper showing that all of the papers.
40,000.
Oh.
Yeah.
I'll take down our fields. I'll have to ask Tom and Anders for some help on that. I'm just kidding. All right, and then here's my contrast. I don't know why I switched to faces and houses. Sorry, this could be positive and neutral or positive and negative. But there's my contrast, 1 minus 1. All right, but remember earlier I said this is just ranking our designs, and we don't want to be picking the best piece of junk out of a pile of junk, because that's still not going to be helpful. The variance inflation factor is nice because it does have a cutoff, a rule of thumb. And what VIF does is it checks for collinearity in your design matrix, and not pairwise collinearity. Pairwise collinearity isn't enough. You can have 2 regressors that combined are correlated to a third regressor, and variance inflation factor will pick that up in your design. So you can just do, what I typically do is I set up a couple of design matrices in my simulation for my loop where I'm going to be iterating and shuffling things. And I just generate a couple, and I check the variance inflation factors of those. If it looks good, then I'm like, okay, I'm going to set this to run 10,000, 20,000 iterations, and I'm going to go to lunch. And then I come back and I pick my best ones. But you want VIF less than 5. Sadly, going back to my story, we had to settle for VIF of 9, which isn't great. High variance inflation factor, if the best you can do is just under 10, your results are still valid, so your type 1 errors are okay. So from my standpoint, I don't care. But what you're going to care about is having a high VIF hurts your power. And you can't have outliers, so you have to look for your outliers. The variance inflation factor causes the estimates to be highly variable. So you're going to lose power if your VIF is high, and you need to be much more diligent about looking at your imaging data for outliers. And you might be thinking, how would I ever find an outlier with a high VIF? Well, lucky for you, I've encountered this before. It is blatantly obvious. So I forget, these VIFs were like 100, but the subject's activation maps were like black and white. That was it. The numbers, the parameter estimates were either like 300 or negative 300, which is what you see when you have really bad VIF. So looking at the data will help you find this. It won't be isolated to a single part of the brain. Like the whole brain's going to just look awful. But, right, so let's look at the code. This actually takes you, it's kind of the same code. Don't go to this. Instead, go to the link where you got the slides. And I have this marked down that I'm going to walk through. Right, and I have just if you just want the R code, you can click on this, and that will take you to a file that just has the R code. Right, so how to do this, we need to create that design matrix, right? And to do that, we need to have something that'll help us convolve with a double gamma HRF. So I'm going to use this library, NeuroSim, because I used to use a different library, but I think this one's slightly more stable. Right, and you all, if you haven't ever used this before, you're going to have to run this line, which I have commented out here, but just run this. It will install it, and then the library will load. Okay, so first I'm just going to walk through making a regressor. Oh, and when comparing efficiencies, it's best if you keep the run length, the number of time points, fixed. Yeah. Anyhow, so first we're just going to make a single regressor, and then we'll build off of that. So I'm going to have 60 TRs. I have to specify the TR. I'm going to assume the TR is 1 second. For this, don't worry if you don't know what the TR will be in the future. You can just use 1 second. That's fine. And here are the onset times. I have 3 trials, one at 10 seconds, or one at 20 seconds, and one at 30 seconds. So this is specified in seconds as well. And this is the duration of each trial in seconds. And this functions actually for simulating fMRI data. And it turns out that, just wondering what JB's writing over there. For simulating data, you would also want to build in an effect size, but we're not actually trying to simulate data with an effect size. So I'm just going to set the effect size to 1. So that basically is saying, please don't scale my regressor, leave it at the height of 1. So just set this to 1. And once you have all those things set, you create your regressor using the specified design function, which is in that library you loaded, and you just feed it onset times, the duration, the run length, the TR, the effect size, and use the double gamma. And then you get your regressor. Any questions about making a regressor? Okay. Right, and I have little things here you can try to do on your own. Yes?
Maybe it was about, but like, do you include jitter or anything?
I haven't done that yet, but it's below. All right, so this section here, I'm redoing that example I showed in the slides where I picked the best and the worst design. So let's see. There's 2, I switched back to faces and houses. Right, so faces and houses, and all I'm going to do is shuffle. I'm not jittering yet. I'm assuming that there's a fixed ISI of 2 seconds, but I'm going to shuffle the order of faces and houses. But we know the most efficient design is going to be all the faces and then all the houses. So that would make me as a statistician very happy, because it's your most efficient design, but it's going to make your subjects very bored. So it's statistically appealing, psychologically it's very unappealing. And there's a big push and pull between these two things as you're designing your studies. I often keep trying to budge, can you have a 2-second ISI? No. Can you, 1.5-second ISI? And you have to compromise between the two things. Okay. So I want to restrict it to only allowing 2 faces or houses in a row. So I have a while loop in here that'll do that. So I'll take you through this code. First I have the number of trials in each class. So this is allowing for 30 faces and 30 houses. And then I have a while loop here that's basically going to randomly order the faces and houses and stop when I get an ordering that doesn't have more than 2 repeats in a row. So I set my keep looking to 1, and my while loop's going to keep looking as long as keep looking is 1. This rep This is just creating a vector of 1s and 2s, 31s, followed by 32s. Sample is shuffling them. So that gives me the random order. I just discovered this RLE function. And what this does is it finds how many repeats you have. So if I pull out the lengths part of this, it tells me how many repeats. So it's basically a way of more efficiently writing a vector of data. So if I have 1, 1, 1, 2, 2, 1, 2, 1, I could write this as 1, 2, 1, 2, 1, and I would need 2 pieces of information. So this is saying 3 repeats of 1, followed by 2 repeats of 2, followed by 1 repeat of 1, 1 repeat of 2, 1 repeat of 1. So this can, that's what this RLE function does. Lengths is the second piece of information. So all I do is I pull out lengths and I check to make sure that the biggest number is 2. So this, if this is greater than 2, that means it's 3 or larger. So if this is true, that's equivalent, to setting keep looking to 1. Anyway. You can kind of exchange a 1 for a true, which is what I'm doing here. So that's what that does. And in this case, it doesn't take too long for it to find a design. If you had fewer trials, it gets more difficult. Then I set up the duration in the intertrial interval. So I'm doing this a little bit different than I did above, so that if you did want to do jittering, you would put, instead of 2s over and over again, you would put the jittered values. And I have code for creating the jittered values at the end of the tutorial. So for example, if you used a uniform distribution, you would sample 60 samples from a uniform distribution and put that vector here instead of a vector of all 2s. Or if you wanted a truncated exponential, you would sample 60 truncated exponentials and put those here. The duration, though, is you're probably not going to vary the duration. Okay, so I'm using this cumulative sum function to create the onset times, because all I have to do is take the duration plus the ITI, and that tells me when my next trial is, right? But it only ever tells me when the next trial is. The first trial, I'm going to assume, starts at 0. So that's what I'm doing here. I'm taking the cumulative sum, I'm adding this 0 because that's when my first trial starts. And then this part is going to tell me when the next trial begins. The only weird thing is it's giving me the next trial after the last trial, which doesn't make sense, because I have a duration for the last trial and an ITI for the last trial, but then we're done. So here I'm trimming off the last trial. If that doesn't make sense, just run this slowly later on and look at it, and hopefully it'll be clear. But that's a way to get your onset times. So it's not split by trial type, and that's okay. I'm going to add that in later. Then I create the run length, which is I just add 9 seconds to the maximum onset time. It's to allow that bold response to be recorded for the last trial. And as above, I set the TR to 1 and the effect size to 1. And then I create my 2 regressors, just as I did above. But I have pulling out the onsets for the first trial type, where trial type was created in that while loop, and the duration for that trial type. And then I just used the 2 for the houses regressor. And then I plot it. So now I've just made one design matrix. I haven't done the efficiency calculation yet, but we're getting there. So now we have 2 regressors, created slightly more complicated than the first regressor we made. Questions about this? All right. Now we're ready for efficiency. So I'm only going to make 10 designs just because it takes a while to run. In reality, you would want to do as many as possible, 10,000. So I'm creating let's see if I remember. Oh, whoops, I didn't compute the efficiency. Sorry, skip that part. Once you have your design matrix and your contrast, you can compute efficiency. So I have to add a column of ones to my design matrix. I didn't show that earlier, but you always have a column of ones. In FSL, it's there, you just don't see it. In SPM, you actually see it. I don't know about AFNI because I have less experience with it, but it's there somewhere. You may see it, you may not see it, but it's being modeled. But here we have to explicitly add it. And so our contrast is 0, 1, minus 1. So that's faces, houses. And so the efficiency, this is just R's horrible matrix notation for this, the reciprocal of this. So the transpose of the contrast matrix multiply by the inverse of transpose the design matrix times the design matrix, and multiply by the contrast. And it's 19. Is that good or bad? I need another design to compare it to. So that's what this is doing. Again, you would definitely want to run like 20,000 or more. And basically taking the code I had above and inserting it into a for loop. But I'm leaving the stuff that doesn't change as I loop outside of the loop. So it's the number of trials, the duration's not going to change. In this case, the ITI won't change. If you're going to use a random ITI, you would move this into the for loop. Onset, and as well as this, the onset time calculation, because it depends on the ITI and the run length. So all of this would have to go in the loop if you were changing the ITI on each iteration. And then we have the TR and the effect size. Importantly, you want to, of course, save the efficiency values, which I'm calling FVal. But importantly, you also want to save the design matrices or the timings and orderings of your stimuli. In this case, I just wanted to show the design matrices, so that's all I'm saving out. So I'm just initializing an array of zeros to store my design matrices. Okay, so here's my while loop from above. That's exactly the same. Creating my faces and houses regressor, creating the design matrix and the contrast, and then I'm estimating the efficiency and sticking it in this vector, and I'm sticking the design matrix from this iteration into my array of desmats. So I ran this just 10 times, and then this is just code to plot the best and the worst. So just out of those 10 iterations, this had the highest efficiency and this had the lowest. And you can see, remember, the blockier things are, the more efficient it will be. And I should look at my screen. Are these the same plot? No. So you can see that the lower efficiency one, this one's not a spectacular example. It has more wiggles in it. When you run it again, it'll look a little bit different, of course. But it has more of these smaller, this is one trial that's isolated. The bigger one is when 2 trials were in a row. So you have, on average, you'll have more instances of 2 trials in a row for the high efficiency, and you'll have fewer for the low efficiency. Anyhow, and I didn't write out the numbers. I should have, but let's see. And last but not least, you want to compute the variance inflation factor. This is the equation for it. You can use functions to compute it, but this is quicker. You take the correlation matrix of your design matrix. So this is a correlation. I dumped the intercept here. So just the correlation matrix between my regressors. So this is a 2 by 2 matrix. Then I invert it and pull out the diagonal. In this case, since I only had 2 regressors, the VIFs will be the same, but they're both less than 5, so I'm good. I have, for on your own, if you want to jitter the ITI, this is how you generate uniform values. If you want to use a uniform distribution, you would replace, let me go up here, this ITI, basically all these lines would have to move into the loop, and you would replace this ITI definition with that RUNIF command that's below. I feel like that is not the best way to jitter. Can't remember which paper. Probably a paper by Tom Liu. Anyone know that the uniform distribution's not a good one to use, that the truncated exponential is better?
Why is it better?
Yeah, probably Tom Liu. But I notice a lot of people when they use, I'm picky. If I use a truncated exponential, I want to know exactly what the theoretical mean is. It's never described this way in papers, because I know they just simulate a bunch of exponential and they dump the ones that fell outside of the boundary. But this code will allow you to know exactly what the theoretical mean is. So the truncated exponential, you have the exponential parameter lambda, the number of samples. T is the max value. And then I have a shift of 2. What am I? M theory plus shift. What am I doing? Oh. Where's the max one? Okay. So the shift, if I don't shift it, if I do a shift of 0, the values are going to range between 0 and, in this case, 4. But I'm going to shift it by 2, so it'll range between 2 and then 6, if that makes sense. So this will give you the theoretical mean of the truncated exponential. This generates the samples, and this will tell you the actual mean, including the shift. So if you want your mean ISI to be 2 seconds, you would choose these parameters until this gave you a 2. Or was close enough to 2 that you were happy. In this case, the parameters give a mean of 3.2 seconds. So the mean ISI will be 3.2 seconds. And in this case, for the sample I generated, it ranges between 2 and 5.3 seconds. So there are tools that do this for you without you having to write so much code. OptSeek's a really popular one. Technically, OptSeek is not designed for convolved regressors. It's for if you're using a finite impulse response function, if you know what that is. And most people don't use that to model their data. But I still know people that used OptSeek and their designs worked really well, so who knows. Another tool is this NeuroPower Tools, which was developed by Yoka Durnez. I'm going to write her name because I think most people think it's pronounced joke, but it's not. So, Yoka Durnez. So her tool is kind of cool because the search you're doing, this random search, isn't necessarily the best way to find the best design. It's not the quickest way. And her tool is based on some work by, I'll think of it later, Nicole Lazar. It wasn't Nicole Lazar. She's a co-author. I'm forgetting the first author's name. But anyway, it does a more clever search. And it allows you to put in more, in a way it's more flexible, in a way it's more restrictive. I don't know if in that tool I could say, please no more than 2 in a row. And often when I've done efficiency calculations for psychologists, none of the tools would work because they had so many constraints that had to hold. So I almost always end up using this code I just showed you to do efficiency calculations. Yeah, so now hopefully you can use the code too. And if you get stuck, you can just email me and ask me questions. Just remind me that I know you from here. I'll be more likely to respond in a timely fashion. Just introduce yourself. I always get fooled by the people who are like, hey Jeanette, I have a question. And I'm like, I must know you. And it's like, I don't know you. So always introduce yourself when you email a stranger. Any questions about this? The fun thing is, if you look at your efficiency range, if the least efficient design, if the most efficient one is twice as efficient as the least, that means you've cut this whole chunk in half. And typically that is what I see. The difference, the ratio between the least and the most efficient out of my search is often around 2. So I don't know, why wouldn't you want to cut your within subject, this chunk, this level 1 total variance component, why wouldn't you want to cut that in half? So, okay, well thanks. Oh, yeah.
So how does that equation change if you want to try to also model item level random effects?
If you want to what?
Model item level random effects?
Item level random effects. So what's, I'm not picturing what the model.
So if you were gonna, so not necessarily formulating it as a step 1, step 2, but if you were gonna analyze like effect plus a random slope and intercept of effect per subject and a random slope and intercept of effect by item.
I guess I'm not following. Sorry. JB's following because he is nodding his head.
It's that was presenting at OHPL.
Oh, the pandas?
Yeah.
Yeah, I missed that talk.
So it's basically there's a variance component at the event by event level. So each event is not like this, you know, they can't be modeled just by a mean of the event and some random noise on top of that. They have and the random noise of
The events over time?
Yeah, so let's say we're presenting a phase and then another phase and then another phase. The magnitude of the effect of each of those events is varying more than that can be predicted by the variance of the noise.
I see.
So there's a variance component associated with those event per event.
So you have to model each event separately first, and then.
Well, I mean, there's ways of modeling. It's a mixed effect model with another additional level.
Right.
Those sigmas become covariance matrices.
But the design matrix is going to be, I mean, it's totally different.
Yeah.
Yes.
Design matrix is gonna be-
Yes.
Because you would have to have each event separately, and then there's.
So yeah,
The one sample
it's a random
t-test.
effect at the level of the.
Yeah.
I mean, yeah, there are ways of. You could say there's one column per event, and then add some constraint on the solving problem. Then you could. There's various ways.
But I still think, I wonder, I feel like the most, even if you stuck to this and did an efficiency calculation, I feel like the most efficient thing in this model would be the most efficient thing in that model. Right? Because it's.
Probably.
Maybe only in expectation. Because you'd have to say like the effects have mean 0 overall, the item-wise effects. So that's like, I'm not from Cogs, I'm from diffusion, so it's straight logic. But in mixed effects models, you usually take the assumption that the random effects are themselves like mean 0.
Oh yeah,
right?
they have to be mean 0.
And so
yeah.
I think she's right in expectation. It might not be true when you get to the actual, once you estimated your random effects, which sounds kind of silly, but once you try and fit them, then you'll have a non-optimal design, but you won't know that until post-hoc, right? So beforehand, in expectation, this will still be best.
But I feel like, but this would affect the quality of the random effect estimates, right? This efficiency would. If I'm understanding, because you have to be able to look at the trials separately, you're not going to be able to. Oh, it's just so different. Yeah, it's almost like asking for people doing
It's random design.
trial-specific MVPA analyses how to have the most powerful design. I mean, it's kind of the same idea because you're looking at them separately.
I think if you want to be able to estimate the fully cross design, so subjects and items, you present each item multiple times to each subject, and so then I guess that would, like, each item that you're presenting would have to go into the design matrix.
Right. So if you presented it 60 times, you'd have 60 regressors, or well, if there's
one regressor per item.
right. So if there's 60, per item or not presentation.
Per item, not per trial.
Representation.
How do you get
Per
it?
item. Right.
How come you don't have to do it per trial.
So if you're estimating an item level effect, so
But how do you get the variance estimate if the variance of that if they're all modeled together?
right at some point you're going to need single trial estimates.
I think the assumption is that some of the faces you're showing are repeats, and that for that specific face, you want to model the effect of that specific face, even though overall you're looking at the class of faces versus cats or something. But
That's correct. Yeah. And then you kind of you have an additional variance in your.
Yep.
You would have the variance of the inter-subject, inter-event.
I'm not even
on
going
top
to.
of it. So
I'm not even going to try to write it because I'll screw it up. This thing would be moved. It would almost be added between these anyway. The design matrix is still at the first level. The second level is basically a one sample t-test. So that anyway.
it's fitted together.
Yeah, I wanted
It's
to.
not fitted arguably.
Yeah.
Maybe a less complex question. I was wondering regarding experimental designs where the design matrix can't be known until after you've observed subject performance, like a self-memory effect.
Yeah.
I know maybe, so first, can you optimize in any way? Second, could you at least use these tools to describe maximum and minimum efficiency of the emergent design?
So I don't recall what he did in the paper, but Ben Turner did a paper that did look into that. And I don't know if he has a method that does allow you to somehow do it, but I know he did look into it. Yeah, he used to be at UCSB. That's just Ben. It's probably Benjamin on the paper. UCSB. At the time he was at UCSB. He just moved to Singapore, I think.